Good morning, everyone. My name is Judith, and I'm from third MSc BT from Department of Life Sciences, Christi Jayanti College. And today I'll be talking about the enzymatic method of DNA sequencing, which is taken under the guidance of Dr. Manikantan. DNA sequencing is a method used to determine the sequence of bases present in a DNA molecule. Sequencing provides information on the order of the nucleotides and its arrangement. It is the arrangement of these nitrogen bases, adenine, thymine, cytosine, and guanine, that is determined. The basic mechanism of this process is that signals are generated and analyzed on the computer by radio labeled or fluorescent labeled nucleotides after amplification. Though there are different methods of DNA sequencing, the steps involved in the process follows a generalized pattern. Talking about the enzymatic method of DNA sequencing is known as chain termination sequencing or Sanger sequencing or dideoxy sequencing. The exact order of a segment of DNA can be obtained by this method. This method was developed in 1977 by Frederick Sanger and his colleagues. DNA fragments of length ranging from a few hundred base pairs to tens and thousands of base pairs or even whole genomes can be sequenced by this method. Only a single piece of the template DNA sample, known as the target DNA, is sequenced in a single sequencing reaction. Plasmid DNA, genomic DNA, bacterial artificial chromosomes, yeast artificial chromosomes, etc., can also act as a source of template DNA. So, the principle of Sanger sequencing. The underlying principle of this method of DNA sequencing is based on the separation by size of target portions of a DNA molecule. Each of the DNA molecules starts at a common 5' end and terminates at one of the many alternative 3' points. So, to determine which nucleotide is incorporated into the chain of nucleotides, 4 dideoxynucleotide triphosphates labeled with a distinct fluorescent dye are used to terminate the synthesis reaction. So compared to DNTPs, DDNTPs has an oxygen atom removed from the ribonucleotide. Hence, these cannot form a link with the next nucleotide. Following the synthesis, the reaction products are loaded into 4 lanes of a single gel, depending on the diverse chain terminating nucleotide, and are subjected to gel electrophoresis. So according to their sizes, the sequence of the DNA is thus determined. So this is a structure of a DDNTP and a DNTP. So here we can see the absence of an oxygen molecule in a DDNTP. So next is the procedure for Sanger sequencing. So the sequencing method consists of 6 steps. So first, the double-stranded DNA is denatured into 2 single-stranded DNA. And then a primer that corresponds to one end of the sequence is attached. The 4 polymerase solutions with 4 types of DNTPs, but only one type of DDNTP are added. Then the DNA synthesis reaction is initiated and the chain extends until a termination nucleotide is randomly incorporated. So the resulting DNA fragments are denatured into single-stranded DNA again. So then the denatured fragments are separated by electrophoresis and the sequence is then determined. So this is an overview of the methods of the Sanger sequencing. First, the double-stranded DNA is denatured and a primer is attached. The 4 solutions are added and then it is allowed to extend until termination. And then again these double strands are again denatured and then electrophoresis is done. So over here, each of the DDNTPs are given a distinct fluorescent label, and when run in an electrophoresis, the DNA sequence can be determined based on the size of each of the single strands. So in conclusion, there are various infinite scopes in various fields of the DNA sequencing, and the major application is to diagnose many genetic disorders. It can also be useful in the pre-implantation and prenatal genetic studies. So apart from the conventional enzymatic and chemical methods of sequencing, there are next generation sequencing methods which are more advanced and reduces time duration, costs and errors that can be used in the modern genetic techniques. So these are my references. Thank you.